All right, thanks. Thank you to all the organizers for inviting me here today. It's a really interesting conference. I think it's been really interesting for me even before I came because as the goal, I'm supposed to deliver a lecture that's oriented towards undergrads on the one hand. On the other hand, I'm expecting all of you to listen through it. That's actually a tall order because you're going to know a lot more about this topic than the average undergrad. And the other is that I had to think really hard about what I can actually present in terms of material, because it's I typically teach computer science undergraduates. I do not teach biology undergraduates. So this lecture is aimed at computer scientists, but not necessarily bioinformaticians. So this can be a lecture, for example, for an algorithms course, in which I try to expose the students to some interesting ideas within bioinformatics in order to draw them into the field. I think we'll have this discussion later as to what extent, how much do we need to teach the biologists bioinformatics? That has been a sort of an axiom from which we started, but sometimes getting rid of axioms makes problems easier. So the way actually this would be is these are two questions which would have come up based on the previous lecture within the class. So the first one is, given two sequences, I'm assuming that basic alignment has been covered. Can we enumerate all of the near optimal alignments between them, where at least optimal or near optimal? And the question is, how many alignments of this type are there? And the second is a classic problem, puzzle problem, which probably many of you are familiar with. There are n gnomes who are standing in a line and looking in a specific direction. Each gnome can see those in front of them, and can hear everybody. And they're all given hats of two colors. Each gnome is allowed to say exactly one word, which is the color of their hat. And how do they agree to maximize the number of hats that they can guess correctly? So how many of you are familiar with this problem, the gnome problem? Okay, yeah, about half of you. Good. So just...
You assume that you can know if it's in front of you or behind.
Yeah, they're just standing all facing in one direction. Like this is the old style communist line where everybody sees just people in front of you. Yeah. So yes, you know who is in front, you know who is behind. Yeah. So this is a very classic puzzle problem. And just to make it a little more fun, because many of you know this already, instead of 2, we can do 4 colors. That makes the problem a little bit more challenging, depending on the solution that you know for 2. And now that all of you have stopped thinking about my lecture and have started thinking about the gnomes, I'm actually, so this should have happened before the lecture even started, I can go on to the actual topic of the lecture. And this lecture is really motivated by high throughput sequencing, and this is the development of these novel technologies that are able to sequence genomes for much cheaper than we were able to in the past. And the way it works in one slide is basically, there's a physical slide on which you attach pieces of DNA, and as they're sequenced, they fluoresce. And this is done through some kind of hybridization of tags, which are then laser activated. And actually, there's a big image which is generated, which are all of these molecules, each one corresponding to a different piece of DNA. And when you look at an image, you just see a whole bunch of colors where each one corresponds to the nucleotide at that position in the corresponding portion of the cell. And then when you do this in many, many cycles over time, you get subsequent letters at that position. So green is a T, then a G, and so on. And similarly, over here you get a different string. And this allows you to sequence lots and lots and lots of DNA sequences in parallel. So, why is this great? Because this enables us to do high throughput whole genome shotgun sequencing, in that we can take a genome, put it through a sequencing machine, and generate a whole bunch of reads. These are pieces from the genome, and they're going to be reasonably short, about 100 nucleotides. There will be lots more errors than some of the other sequencing, older sequencing technologies, but lots cheaper. So these can be used for discovery of variation amongst populations, for figuring out how cancer cells differ from healthy cells and other topics. But the key step which is required to enable all of this is to find for each read its proper location on the genome. So this is the problem of mapping the read or aligning the read to the genome. So, how does this work? There are, again, many methods which I do not go into in any detail, besides to explain that the basic idea is that we have reads, we have the genome. We're going to look for exact matches. How do we go about doing this? There are, many ways. I probably, at this level, would not talk about suffix arrays even to mention them. Just say, look, you're going to find exact matches. I sometimes will show this very basic seed table idea. And then to say, look, there are going to be areas where there are matches between the reads and the genome, if these are exact Hamer matches. And for all of these areas, you're going to have to do an accurate analysis of them to do full dynamic programming solution of using the Smith-Waterman algorithm. And then you can sort of shift along the genome, find other areas which match, and then do the full alignment. So basically, the problem is that you find some candidate regions, and now you have to do accurate alignment within these regions. But we know how to do accurate alignment of two strings. What happens when the data doesn't really look like strings anymore? What can we do? So one particular application of this idea is the mapping of amplification-based sequencing versus single molecule sequencing. So with amplification-based sequences, basically there's an underlying step where we take every single piece of DNA and make many copies of it. And the light that you see when you fluoresce is the sum of all of these signals. So this is going to be much more accurate in terms of getting the right signal, but there could be some systematic errors. So if there is an error which happens during amplification, you may end up seeing that the error base instead of the correct one. With this, there are many more sequencing errors, and these sequencing errors have specific types which are actually going to be interesting. You're more likely to skip a letter or to clip the ends, and what some SMS machines do for this is two-pass sequencing of the reads. So you have this original sequence, and you actually generate two separate reads. One which maybe missed some letters, and another one which missed some of the other letters. So the question is, how do we align these two? How do we figure out the correct source of two of these reads? And where do they map onto the reference genome? And of course, the correct way to do this is to do three-way alignment of the sequences. So the same way you can do dynamic programming in 2 dimensions using the Smith-Waterman algorithm, you can do dynamic programming in 3 dimensions, where each cell now going to depend not on 3 previous cells, but on 7, if you think of the cube previous cells. And you can do the computation. But this is going to be way too slow. This algorithm is going to take running time, which is the product of the sequence lengths, not, and for 3 sequences, this is obviously unacceptably slow. The other option, you could try to map the reads separately. But if you look at just one of them, you require just one of them to match. It could be that there are too many missed hits. That there are too many potential matches. If you require that both of these match in the same location, it could be that you've completely, that you don't get a hit from both of the reads because each one of them is reasonably low quality. So what you really would like is a solution which takes advantage of the information that you know these 2 reads should be coming from the same area and takes advantage of the similarity between them, but does not have the running time of the cubic running time. And here is where, if you remember, of the 2 questions which I asked in previous lecture, the first one comes in. So if I think about the 2 reads. I could align the two of them to each other and then generate a profile. Basically, whichever letter, pick the one letter from each one of these and say, okay, well, this is the new sequence. This is what I should be mapping to the genome. Unfortunately, so before I go on, you should notice that the scores I'm using are really, really weird in that the gaps are very cheap. And this is because within this technology, gaps are actually the preponderant type of error. So if it's something common error, you want to have a very low penalty for it. So if this is the alignment, this is the profile, and you could try to align this to the genome. But there's actually a second alignment which is just as good for these two sequences. You could align them this way, and it has the exact same score. So this is the idea of there are two co-optimal alignments. And the question, and this is what you would have had to figure out based on the homework question from the previous lecture. Similarly, there are actually 4 more near optimal alignments, which are just one score of 1 less. So this is 9, and these two dark green ones are 8. So there are additional paths, and which give you this score. So there are these near optimal alignments. You can see that the number starts exploding exponentially. So you don't really want to enumerate all of these sequences and then try to align each one of them. But what you can do is you can actually represent all of them together and align the whole set. And here comes an idea. So first I'll show you how to compute this set and how to represent it efficiently. And here what you have to do is use a trick, which is use the Smith-Waterman algorithm not just forwards but also backwards. And here's the trick. To find all the near optimal alignments, we first compute the regular dynamic programming matrix the same way as you would have done it previously, and this gives you sort of the score of the best alignment up to a certain cell. So the best alignment up to this cell has a score of 7. Up to this cell has a score of 4. Up to this cell has a score of 0. But then you also compute the best alignment. You do the exact same computation, but you go backwards. So this gives you the best alignment to this cell, but coming from the end. So this would have a score of 4. The best alignment to here would have a score of 5. Here 8, and so on. By adding the two alignments together, you get actually, for each cell, the score of the best alignment that goes through this cell. And this trick gives you that these are the cells through which the best alignment could go. Now, of course, the number of potential paths is exponential, but the number of cells is going to be at most quadratic, but in practice much, much, much smaller. And here is, once you have this, you can actually take all of these cells and represent them, once you remove the really suboptimal ones, represent them as something called a weighted sequence graph, which is basically a DAG, which describes all of these cells and how you can go between all of them. So if you think about this, this DAG represents all of the possible near optimal alignments between this. So that's great, but what can we do with the DAG, right? This is, it's a representation of all of these alignments, but we now need to align, all of these. Do we again start walking these and aligning each walk to the reference? Well, it turns out you can do something smarter with these weighted sequence graphs. One thing that you can do is actually think of a string as just a linked list. And you would think that, why is this crazy? You're making things more complicated. We used to have something simple, and now you have this graph. which of these of the individual letters. But it actually turns out that by thinking of it this way, you can start thinking of the dynamic programming Smith-Waterman, Needleman-Wunsch family of algorithms in a somewhat different way as well. We can start thinking of them as we have one sequence along one axis, we have another sequence along the other axis, and really what you have in between is just the cross product of these two graphs. So for each of these graphs, I've made a copy of it and have filled in the other half with the other nucleotide. This is identical to regular Needleman-Wunsch, but now we're thinking of this as a graph. And really all that Needleman-Wunsch algorithm does is it actually finds the longest path through this graph, where you have certain penalties on, edges, depending on whether the letters match or whether they're vertical or diagonal, vertical and horizontal or diagonal. And actually, if you think about affine gaps, for example, you can also represent those as a graph and finding a longest path within this graph. So why is this useful? This formulation? Because actually nothing that I've done here assumes anything about this graph besides the fact that it's acyclic. And if I have this weird weighted sequence graph, well, I can make two copies of it, and I can connect them all in the exact same way, allowing for and connect them all and compute the longest path through this cross product graph. So you can align the weighted sequence graph to a genome, or actually to another weighted sequence graph, in time that's proportional to the product of the graph sizes.
Why is it called weighted?
Because it's called weighted because each of these edges is going to have a different weight depending on the score on the previous of the alignment in the previous level. So this idea allows you to find not just a single alignment, but many, and align all of them in reasonable time at further levels to the genome, which is on this axis. So the take-home points from this section are that, well, alignments are just graphs. And this, there are some questions which arise. First is, how can we solve the longest path problem on these graphs? Shouldn't that be NP-hard? Obviously it's a take-home question, but then how can we enumerate all the near optimal alignments? If there's an exponential number of them, why are we able to enumerate their set and work with it, but actually can't enumerate all of them in a reasonable amount of time? And the sort of a more complicated question, which actually requires quite a bit of thinking, is can we convert the longest path problem to shortest path and then run Dijkstra's? So this assumes that the people are familiar with Dijkstra's algorithm. If we can, would it be faster? And it's actually, this question is, I wouldn't be surprised if many here don't know would need to think about these before they're able to figure it out. It's very non-trivial. Why Dijkstra, how to convert, you can, but it's, there's one trick that you have to do. And running Dijkstra's would, in the worst case, be slower, but in many cases actually is faster. In many practical cases is faster. So it's, these are interesting sort of questions to take home. The second part that I want to talk about, I can show as an application of this, is this idea of dye-based sequencing. And this other sequencing technologies, instead of sequencing, and this is specifically the solid technology by Life, instead of sequencing individual letters, sequences pairs of letters, which is why it's called dye-based. And what you get is a signal which is the sum, which is the sort of, it's based on the two letters around it. And if the two letters agree, for example, A, C, and C, G, and G, T, and T, you get a 0. And in other cases, you get 3 other colors. So there's a total of 4 possible colors which the technology could generate. But each color doesn't correspond to a base. It corresponds to a pair of bases. And the code is lossy in that multiple pairs give you the same signal.
So it doesn't distinguish between AA and CC?
Yes. So if you get a string of all zeros, it could be all A's, all C's, all G's, or all T's. If you get a string 0, 1, 2, 3, it could be A, C, G, T, or C. Well, there will be 4 other combinations which you get. So how can we adapt sequence alignment algorithms to deal with something like this? This is a, and the key, it turns out, instead of thinking about these transition matrices, like these pairs, we are computer scientists. Yep.
So it's missing.
What do you mean?
Because the number of distinct tables is 4. You have only 1 to 3. So you need the AC distinguishing, AG, AT, and CG, CT.
What do you mean it's missing one?
Can you distinguish every pair of distinct letters? Do you get a new number?
No. There are 16 possible pairs because these are ordered pairs. Well, not 16, but 12, we could guess, because you have to. But you get only 4 colors. You get only 4 colors: 0, 1, 2, and 3. So the code is quite lossy. But the question is, what can we reconstruct from it? And the way it turns out, an easier way to think about this, and this goes back to your sort of the question of whether it's how best to think about it and how it's lossy, is instead of thinking as this is actually the transition matrix of a graph, and this is the corresponding finite state automata. So you have a nucleotide, and depending on which of the 4 colors you observe, you go to a different nucleotide. From each nucleotide, you can get any of the 4 colors, and it'll always take you to a different next nucleotide.
So 3 confuses between 2, AT and CG.
What do you mean?
3 is the same for, okay.
3 is the same for AT, TA, CG, and GC, those 4 combinations. So it's interesting. Exactly. This is why it's interesting. That's my goal when I'm teaching this, to show the students that it's interesting.
Sorry, if you know the first one for sure, then you for sure know the whole sequence, right?
Exactly. So this is the other thing that becomes very obvious when you see this as a finite state automaton. If you start at a specific location, then all of the other ones are going to be deterministic. But if you actually just look at this, it's not as obvious. That's a very good point. So what can we do with this? How do we align colors? And one thing is, you do need to know the first color. You need to know where to start. And the other is, how does this code sensitive to things like changes in the DNA? What if one of the letters was changed? How would this code change? What if a letter was inserted or deleted? How would the code change? If I had a sequencing error, which means I misread one of the colors, what does it mean at the level of the DNA? It actually turns out to be far from trivial. So the way some of the first programs approached this is they actually would take the genome and convert it to color space, because that's easy. You take the genome, you convert that to color space, and now you look at the read, and you look at one of the things they said, oh, if you see 2 colors which are in the row different, that actually corresponds to a SNP, to a single nucleotide change. So here you can see a G changed to a C, and actually that changed 2 colors. And if it's only 1 color that changed, that's a sequencing error. But that's actually not completely true, because how many ways are there to change 2 colors? As Soren pointed out, there are about 12 of them. Yeah? But how many SNPs are there? 3. So there's something going on. So there are 4 possible letters, and that will give you 4 different code. These are 4 options. What happens if the 2 of the 2 change, but instead of from 2-1 you go to a 1-3? What would that mean in terms of the sequence? That's actually not going to be a SNP. And it actually gets much trickier when you start looking at indels. If you start with this sequence, and then you have one which deletes a letter, what happens is you delete a color, but you also change the adjacent color, because the previous letter has changed. Similarly, you can insert a letter, and if it's actually similar as the letter next to it, you just insert a 0. But if it's different than the letters around it, you not only insert, but you also change an adjacent color.
When you have a deletion,
How
how do you do the pair coding?
do you?
Do the pair coding, because you basically miss that.
Right, so it's relative to the previous one, to the one right before the deletion. Because the coding, right, when you're sequencing, you know nothing about insertions or deletions. You just code this string. But when you map is when you realize, oh, there is a gap there. So this is this is why the 3 changes, well, why the 1 changes to a 3, because the 1 was relative to a letter that has been deleted. So it becomes very tricky to compare these sequences in color space. You're looking at things, some things change. Finding a distance metric is going to be very, very tricky. So by the way, has anybody figured out what the pattern is for SNPs? What is the rule? Sorry? Some must stay the same. Yes. So this actually, some must not quite stay the same. Some modulo 4 should stay the same, and the other way to represent that is XOR. The XOR has to be 0. That sort of becomes trickier. So, how long are the? So I mean that's so now they're about 35 pairs or 50 pairs. You know, that obviously changes depending on the day of the week, and then as new versions of the technology go out. It's a pair. Yeah. They do both paired end and mate pairs, so depending on how you do the construction. But yeah. So the idea is, so you have these colors, and how do we align these? How do we align sets of colors? So the idea is, okay, let's do, why make it so complicated? Why not do it in letter space? Why not actually take the read, take it from colors and convert it to letters? One of the things I didn't tell you is that for a read, we actually always start from the specific letter. We insert a fake letter before it so that we can actually know where we start. So the idea that if this was the read, and this is how you would go about translating it, but unfortunately, as soon as you reach to a sequencing error, all the letters become wrong. So if this 2 is misread, if this 2 is wrong, all the subsequent letters are screwed up. Which is why people did not want to do this based on the letter space. It's only half the read matches, or only the portion up to the first sequencing error actually going to match. But here is something that we noticed, and this is actually going to be obvious once you go back and think about the finite state automata. Heck, if I started from a different letter, all the letters up to that error are going to be wrong, but afterwards they're all going to be right. Because, well, what does a sequencing error mean? Well, I just jumped to the wrong position. But because the path was deterministic, depending on where you started, if I started from a different location, there was some position, there was some way of going so that on that erroneous letter, I would jump to the right one, and then everything else is deterministic. everything else is going to be right. So this sort of should give you start thinking about ideas about how to do the alignment actually in letter space. If you think about the sequence, you have 4 different translations, 4 different ways of parsing the sequence. And if all the reads begin with a T, this is the right translation. But after a sequencing error, you jump to a different translation. And this becomes something that you can already start thinking about an algorithm for. There are 4 unique translations, and you can do Smith-Waterman, where you're doing 4 Smith-Watermans at the same time, but allowing yourself to jump between the various Smith-Watermans. So do an alignment up to a certain amount in one frame, but then do it in this frame afterwards, and you have a penalty for jumping between frames. And this actually, if you think about this matrix, this will correspond to the following dynamic programming problem. So you start with these. On a 3, you get, from an A, you get a T, from a C, a G, and so on. So you get these 4 possible letters, and then after that, these 4 possible letters. So to compute the cells here, well, there are the standard Smith-Waterman recurrences. Each cell will, you can go to the corresponding cell in the previous matrix. So these 4 connections correspond to the 4 matches in standard Smith-Waterman, where this cell inherits from this cell, this one from this one from here, and this one from here. But these are similarly the gap penalties and the other gap penalties. These are your regular Smith-Waterman transitions. But you also have the ability for this cell not only to come from here, but also to come from these other 3 cells, but charge yourself a penalty for shifting frames. So this is a sequencing error. You have to jump to a different frame. And similarly, there are 4 possible gaps penalties because you could have which allow for a sequencing error vertically. On the horizontal side, however, you only have 1 recurrence because you cannot have a sequencing error in the genome that's color space related. So in this direction, you only have for a single recurrence. So the take-home points from this are, well, color space actually is also a graph. And the questions are sort of, first, what does color space have to do with gnomes? So do any of you know the solution to the gnomes with 4 colored heads? No? I won't give it away, but think about that. There's actually a relationship between gnomes with 4 colored heads and color space. And the alignment that I show for color space can actually be represented just as an operation on graphs, similar to what I showed in the first portion of the lecture. And this is a question of how to actually do this. And the final, again, it's a very tricky question. Color space reads have the same information content as letter space reads for a given length. So if you have 50 base pair color space read, 50 base pair letter space read, all of them have the exact same information content from like Shannon entropy, if you wish to think about it this way, or just from the terms of the number of possibilities. But for the same length and error rate, if the 2 technologies had exactly the same length and exactly the same error profile, you would actually be able to map a larger fraction of color space reads to the genome uniquely. Why? So this is it's a question which really requires you to think about how the color space representation of the data differs from letter space. And actually, and the hint for this question is, if your genome was just a random sequence of letters, this would not be the case. So.
But you assume that the reads are somehow randomly sampled from the genome.
So you assume the reads are randomly sampled from the genome, but that the genome is actually a real genome and not, you know, a Bernoulli sequence of nucleotides. Because as soon as it becomes a Bernoulli sequence of nucleotides, this claim is no longer true. So that's the sort of, these are sort of the take-home points from the second portion of this class. So, and because this is supposed to be similar to a lecture, I don't have an acknowledgement slide, and I don't thank my funders. I do thank you for your attention.
How is this related to PacBio?
PacBio is an SMS technology. So the first portion is somewhat related. In PacBio, they actually do this weird thing where they go through many, many times through the circularized sequence. So they don't do two pass. The actual work, our own work, which motivated this two pass, the weighted sequence graph idea, using it for high throughput sequencing, was done for helicose, which doesn't exist anymore. PacBio has the similar issues in terms of the error rate, that it's high in terms of indels, and many of the same concepts would apply.
So they talk about 20% error rate. It's of the type you described in the first lecture?
Yep, mostly indel, almost exclusively indel. Yeah. Sorry.
Are your alignment graphs all DAGs?
Sorry?
Are your alignment graphs you talked about in the first half, are these all DAGs?
Yes. So.
I guess I'm a little confused then because we talked about using Dexter's algorithm. The longest path of a DAG is known to be linear.
Define linear. Linear in the graph size.
Linear in number of vertices.
Exactly.
But you built it. So if you're building it, you're stuck with that anyway.
Right. So it's linear in the number of, well, vertices and edges. But so there is the difference between theory and practice, right? This was the question asked about Dijkstra's. So it turns out that in the worst case, yes, Dijkstra's will be worse. But if your sequences are really, really similar to each other, Dijkstra's just shoots down to the right, down the diagonal to the right location.
Okay, so you're
Because it's Dijkstra's, right? It does not. As soon as if it goes straight to the goal, it does not expand any of the other nodes. There's the whole. So. Yeah, so that's where the question of when is Dijkstra's better. It turns out for really similar sequences, Dijkstra's is better.
The statement about Dijkstra needs to be a little bit more carefully done because there are various data structures that you can have Dijkstra.
That's true. That's a good point.
The question should be an implementation of Dijkstra.
With binary heaps and E-log V running time, yes. That has nothing to do with that. You can build the queue in real time.
Yeah, they're just about all the textbook ones, for reasons unknown to me anyway. For some reason, always start by putting everything in the queue for sure.
That, yes, of course, yeah.
So, I try to understand why you add up the forward dynamic programming and the backward dynamic programming. That's the forward.
So the idea, why do you add up the backward dynamic programming and the forward dynamic programming? So because this was a real class, you should have asked me that when I showed it. But let's see, where's that? What's this? So when I do a forward, the standard Smith-Waterman dynamic programming, what I'm really computing is the score of the best alignment up to a certain location, right? So what's the best alignment that goes through this specific cell? Well, you know, it goes somehow to this location, then somehow from this location to right here. That's going to be tricky to compute, but I can easily compute the forward ways that gives me the score up to here. And then, I compute the same thing but backwards, and this will give me the best path, the score of the best path to this cell. So once I add them together, I get the score of the best alignment through this point.
But that's one possibility, right? There are other possibilities. You have maybe two sub-optimal.
So because alignments are additive. So if you chop an alignment, and unless you have affine gaps, which make things a little bit trickier, but if you take an alignment without affine gaps and cut it in half, the score of the whole alignment is equal to the score of the left half plus the score of the right half. So if the optimal alignment up to this point and the optimal alignment from this point on is going to be the optimal alignment through the point. So, but that's, I think it's a good point. I should have emphasized that a lot more than I did in my lecture.
Is the known answer n minus 1?
Sorry?
Is the answer to the known
for both 2 hats or 4 hats.
Yeah.
Yes.
Regarding the color technique, it seems like the disadvantages of using this kind of sequencing are obvious. Is there any good reasons why people would use that sequencing technique rather than this other more substantive one?
There are theoretically good reasons. I think one of the things you have to do is separate the theory and the practice of this. So the actual technology, I think, has been seen as a flop. I mean, not a total flop, but it's basically lost out, and the Illumina versus solid war has been clearly won by Illumina, and very few people actually use solids. But that has a lot to do with the technical issues and with the error rate. If you had identical error rates on the colors and the letters, the colors would be better. So, as I pointed out, there was the issue of the fact that more reads can be mapped uniquely. But this also means that if you have just a single read, which is covering a genomic location, you can figure out if there was a SNP or if it's a sequencing error. Because SNPs and sequencing errors have very different profiles. Right? So this is important because when you have one read in Illumina, a regular letter space, which disagrees with a reference, you don't know if you just got that read had this error in it. So you need lots and lots and lots of coverage before you're able, at least 2, but preferably more before you're able to make a call. Here, because sequencing errors look so different, if you know that this is the right location for the read, you can actually make a SNP call just from one read, in theory. In practice, unfortunately, things aren't as nice. So there are advantages to color space. It's actually extremely, extremely, extremely elegant. It's just the practice of it has been somewhat screwed up.
3 questions, actually. First, all about color space. The first one is, do the quality scores change anything? And are there any tools that seriously incorporate them? Second one, what about the novel assembly with color space? And third one is anybody still buying solid machines?
So I think the answer to 3 is very few people, if any. For 1 and 2, sorry? Russians, well, some Canadians do as well, but that's a different question. But I think it's a problem. I mean, very few people buy them, and for good reasons. But in practice, so this is.
Is there anybody who would specifically buy a solid machine, even though knowing all the disadvantages?
Yeah, it's cheaper. Yeah, it's cheaper. So they're not quite giving them away, but close to that. So. The other things which you asked about are, obviously beyond this sort of the topic of the lecture. But yes, there are quality scores. They basically would change your transition, the error penalty, the probability of jumping between the different frames of the alignment. And there are at least, our tool for color space handles them, and I think a couple of others do as well. So they're being used. And de novo assembly, there are tools specifically for de novo assembly for color space. How exactly they work would be actually a really cool topic for a different lecture, because it turns out that once you build a scaffold, once you do the overlap and layout of the color space reads, figuring out what the actual nucleotides are at each position actually turns out to be as simple as doing a forward-backward in a very clean HMM. But that's sort of beyond the topic of this.
Thank the speaker again.